Hi students, today we are going to talk about Ames test. What is a Ames test? An Ames test is a test which is mainly used to detect mutations. But we should know that when we go to look the number of carcinogens, okay, if you conduct a study, in a study which has been conducted in a book, An Introduction to Genetic Analysis in 7th edition, it has been shown that mutagenicity and carcinogenicity are being found to be clearly correlated. And it has been found that 157 of the 175 known carcinogens are being found to be mutagens. So, when you select or when you study a carcinogen, most of them, approximately 90% of them, are being found to be mutagens. So, considering this, it was being found that in 1970s, a scientist called Bruce Ames, okay, he tried to find out the same policy, that is, many carcinogens are also mutagens. And keeping this in mind, he developed a test called the Ames test. And this test determines if a substance will increase the rate of mutation, that is, if it is a mutagen. And if the substance is a mutagen, then it is also likely to be carcinogenic if an animal is exposed to it at sufficient level. So, by directly checking whether a substance is mutagen, they give a chance that probably it could be carcinogenic also. So, based upon this, the Ames test was being developed in 1970s, aiming to find out the mutation rates or the mutagenic property of a substance. So, let's go into the details of this. Here we basically use some bacteria to test whether the particular chemical can cause mutations in the DNA of the test organism. It is a biological assay that is formally used to assess the mutagenic potential of the chemical compounds, and a positive result from the test will indicate that the chemical is mutagenic and therefore it may act as a carcinogen, as cancer is often linked to mutation. Now, this test has been found to be quick and convenient to estimate the carcinogenic potential of a compound, because the standard carcinogen assays on mice and rats are time consuming and expensive. But of course, you do get some false positives and false negatives also. So, I guess I need to explain these points in some details. Now, if you have a particular chemical which you come across, every chemical which is being used now in your cosmetics or in different industries, or let it be in your toothpaste and everything, all these chemicals have to first pass a test whether that is a cancer causing agent or is it a mutagen. So, and this process usually to check whether it is a carcinogen is usually be done by in labs. We do it like you inoculate the chemical into the animal, into the experimental mice, and then you see whether the cancer development is happening or not. Now, that process of inoculating each and every chemical onto a mice or a rat has been found to be time consuming, and it is also been found to be expensive, because always you have to do triplicates of each experiment, okay, and so this would result in a lot of expense for such experiments. So, in order to bypass this extra expense, we will do the mutation analysis on certain bacteria rather than going directly onto the rats or the mice. So, these bacteria are being first checked that they are being inoculated with the the chemical of interest, and we check that whether they are being found to be mutagenic or not. If they act as mutagenic, then there is a chance that they may be a carcinogen, because cancer is always linked to mutation. So, basically, rather than directly going into the mice and the rats, we take some bacteria and check whether the chemical can cause mutations in the DNA of the test organism. And you can't take each and every microbe that you like. There are certain microbes which are being used specifically for the Ames test. The tester strain which we consider here is Salmonella enterica serovar typhimurium. Now, each tester strain has a different mutation in the histidine biosynthesis operon and therefore is a histidine auxotrop. So, you do remember what is an auxotrop. A histidine auxotrop is something which cannot synthesize histidine by its own biosynthetic machinery, and so if that particular strain has to be grown, you have to supplement histidine in its media. And so the Salmonella enterica serovar typhimurium strain which has been taken here is an histidine auxotrop, and this bacteria is also being mutated in their cell walls to make them more permeable to the test substances. And also to further increase the assay sensitivity, the strains are defective in their ability to repair DNA. So, three important characteristics are being incorporated into the Salmonella enterica serovar typhimurium. Now, you might have an imagination from where do you get this particular strain. These strains are being they are being created in the lab after inducing mutations in them. So, the Salmonella enterica serovar typhimurium which has been used in the Ames test is being found to be a histidine auxotrop. Secondly, the cell walls of this bacteria are being found to be mutated so that every chemical becomes permeable to the cell wall of this culture or this bacteria becomes permeable to every test substance which we are going to check. Thirdly, what happens? You also make the repair mechanism of the bacteria to be found to be as defective. You give a mutation so that the bacteria will lose its ability to repair any DNA defects or any mutations which be induced by the presence of a chemical. Now, what is the protocol? In the Ames test, what happens? These tester strains of Salmonella typhimurium, they are plated with the substance being tested, and the number of visible colonies that are being formed are being determined. And to ensure that the DNA replication can take place in the presence of a potential mutagen, the bacteria and the test substance are mixed in dilute molten top agar to which a trace of histidine has been added. And the molten mix is then poured on top of the minimal agar plates and incubated to about 2 to 3 days at 37 degrees centigrade. So, let's go to a figure so that it's more clear. Now, if you have a culture of Salmonella histidine auxotrop, this is the test strain that we have. What you do is, you take this culture and you are putting it onto two different medias. One is a minimal media plus a small amount of histidine. The second one is a minimal media with a small amount of histidine, as well as you also have the test mutagen, that is the chemical which we want to analyze whether it is a mutagen or not. So, you take two different plates and onto both the plates you take the auxotrop, Salmonella auxotrop, and plate it. But in one plate, one culture plate that we have considered here, we have the test mutagen also been inoculated along with the culture, and we have used a small amount of histidine. We had mentioned the reason earlier, that is, it is mainly to promote that the DNA replication and bacterial growth is permittable to some extent at least. And then you incubate it at 37 degrees centigrade, and what happens? Sometimes what happens? It is a histidine auxotrop. Now, this media contains small amount of histidine, and a spontaneous revertance can grow here. That is, spontaneously itself, sometimes the auxotropic organisms can be reverted back into his positive strains. So that is which has been depicted in this particular plate. But sometimes what happens, under the influence of this mutagen, the auxotropic nature of Salmonella gets reverted back, and you get more amount of bacterial colonies on the plate to indicating that the reversion of the histidine auxotropy has been happened. So, if you have more amount of bacteria formed on the plate where the mutagen has been present, it indicates that that mutagen is really mutating the bacteria. Of course, when I say mutagen, it will be mutagen, but when you don't know whether it is a mutagen or not, we can say that the chemical which we incorporated in that media is being found to be a mutagen because it is causing the shift of histidine oxytropes on this particular plate. Now, this is a randomly occurring. In the case of spontaneous revertance, there is this randomly occurring process, but not under the influence of any mutagen. So, coming to the interpretation, all of the histidine oxytropes grow for the first few hours in the presence of the test component until the histidine is depleted. And this is necessary because replication is required for the development of mutation. And this initial growth does not produce any visible colony over there. But once the histidine supply is exhausted, only revertants that have mutationally regained the ability to synthesize histidine will grow and produce the visible colonies. And these colonies need only be counted and compared to controls to estimate the relative mutagenicity of the compound. The more the colonies on the plate which contains the chemical, then it indicates that the greater is the mutagenicity. Now, this is a picture of the same thing which is I have in biurofins. Here the strain is there, and then you are incorporating in one plate the mutagen, in the other one you do not have the mutagen, and you can see here they have been many colonies are produced by salmonella that has mutated to restore the histidine gene. Now, we will be talking about the remaining portion. Why do we have to use sometimes rat extracts for the Ames test in the coming sessions? Thank you.